Hello and welcome to lecture 5. This lecture has two topics, really. One of them is biofilms and the other is cellular energetics. One of those topics, well, they're equally important, really, but I don't have much for you to learn about biofilms. They are very important, but they're a lot easier to understand than cellular energetics. So the vast majority of these slides and videos will be about energetics. And that is just what is necessary to understand it. So this first video will be about biofilms. So let's get into that. Let's see if this can work. Okay, biofilms. So most bacteria don't live in the water, and they try to colonize solid surfaces that water occasionally flows across. Because remember, they need water. It's the main thing they need. So they tend to stick to things. And what they do is they make this structure that they can live in. And it allows them to easily attach. As you'll see, some of the bacteria do the attaching and others just live there. It protects them from things that might eat them. So the structures they make are much bigger than a cell that might try to eat bacteria. And it also allows them to live together and work together in certain ways to do things as a group that might not make sense to do as an individual. So here are two images you can see. So first of all, let's look here, because this is the formation of biofilms. This is how it works. Initially, there are individual cells swimming around. So let's assume this is water and like a rock in a stream or a river with water flowing over it. So free swimming, or what we call planktonic bacteria, will swim nearby, and some of them might decide to attach. And so if they do, they will start to adhere to the surface. They will use fimbriae and other structures to attach to the surface. And if they're capable of forming a biofilm, once they realize there are enough of them there, they will start to excrete this thick polysaccharide matrix. So it's a gel. And they start to excrete this and then divide. So they make lots of daughter cells, and the daughter cells are sort of stuck in this. And so what that means is that the daughter cells don't need to put any energy into attaching. They just need to divide and make this polysaccharide. And so over time it builds up and gets thicker, and it reaches kind of a maximum thickness. And what we find is that there are holes in it and pores and channels water can flow through. So any cell in the biofilm, it may be embedded in this gel, but it's never very far from the water that's flowing through. So the beauty of this to the cells is that it lets them kind of increase the surface area that they colonize. So instead of being limited to the surface of the solid structure, now a bunch of them can live up here, but they stay in one place while the water flows through. So if there are nutrients in the water and that's what they're using, those nutrients come to them. If they're generating waste products that need to go away, those waste products flow away. And so they can keep dividing and making daughter cells. And once in a while, as this expands, part of it will just break off and float away, and that kind of limits how tall these things get. The taller they get, the more exposed they are to shear forces from the fluid. So typically a biofilm would be on the order of 50 micrometers thick. So that's 1/20th of a millimeter. And eventually the cells would kind of get too abundant, and some of them would realize they're starting to starve because they just compete too much for resources. And so they would switch back into this form. It's not that they're like changing their form, it's that they would change gene expression. So they would go from happily living here to making flagella so that they can swim through the water, whereas these don't need those flagella. These don't need to swim, so they don't make those structures. And there are other behavioral changes that happen for these cells that swim away. So cells that decide to become planktonic do behave very differently from cells that are in a biofilm. So that's kind of the process and the logic behind it. This is a fluorescent microscopy photo of a biofilm. And in this we can see kind of the net-like structure with the big holes. And we can see the polysaccharide has stained blue, and the cells inside it are stained with some kind of dye that makes them a slightly different shade of blue. So it just looks like this big mess, this big net of cells. And so I mentioned that cells typically don't live in water. I mean, there are lots of bacteria in water. There are enormous quantities of bacteria in the ocean. But on land, most bacteria live on surfaces. And so it's fair to say that most bacteria would be living in something like this, or some kind of biofilm. Now, we don't always perceive it, but there are some very clear situations where we bump into those biofilms, and we do perceive it. One of them is on our teeth. So while you're asleep, dental biofilms form on your teeth. And these are not particularly hazardous. So the bacteria that colonize enamel and start growing, those don't hurt teeth. But if we don't remove the biofilm, if it accumulates, then other bacteria can colonize it, and some of those other bacteria are the ones that will release acids that degrade enamel and cause cavity formation, or even gum disease. So some of the late colonizers are the ones that can irritate the gums. Yeah, and so this picture, what it's showing is a dye that people can chew on that stains bacteria. And it might be crystal violet. I'm not sure what it is, but it's useful for teaching people how to brush their teeth. And then this is a photo micrograph. So this is a photograph made through a scanning electron microscope, where cells are these little tiny things. That's a cell. That's a cell. But most of the biofilm is the matrix. And for the point of view of healthcare, biofilms are critically important. So they protect the bacteria from phagocytes in the immune system. They protect the bacteria from antibodies. They actually protect bacteria from antibiotics. So if you have a biofilm growing on, say, a heart valve during bacterial endocarditis, those bacteria are very difficult to treat. The immune system can't do anything about it, and they're safe against antibiotics. So that's one of the worst situations we get with biofilms, is thick growth on a damaged heart valve. And sometimes that has to be removed surgically. But also anyone who has a catheter or any other indwelling medical device for a long period of time is at risk of a biofilm forming on that device. And it can slowly creep into or spread along the device into internal tissues. So the other place where you would likely find a biofilm, anytime you're in a lake or a river and you pick up a rock and it's smooth and slimy, well, that's a biofilm that's formed on it. So if you don't see any obvious growth on it, it's not green, it's just smooth and slippery, that's because a biofilm has grown on it and it's very thin. Okay, well, that's biofilms. That's plenty about biofilms. I'll see you at the next video.